Me get started then. All right, so yeah, so it's a pleasure. We're sort of continuing a non-invasive interrogation of the brain theme following Ben Hamstead's talk last week. So it's my pleasure to introduce Mike Vesia from the School of Kinesiology. Mike's assistant professor in kinesiology. He is an expatriate Canadian, trained actually mainly in Toronto, I think. He has a PhD from York and did postdoctoral work at the University of Waterloo, and also I think with Robert Chen in Toronto as well. So those of you who may or may not know Robert is a leading DBS investigator, Parkinson's disease specialist. Mike's interest is in various aspects of motor control, both in normal function disease, and is particularly interested in using non-invasive methods to try and interrogate the motor system. And he's going to talk to us today about some of the work he's done on trying to manipulate these brain circuits with transcranial magnetic stimulation. So Mike, thanks very much for your participation today.
Well, thank you for the invitation, and thank you, Nancy, for organizing all this. Let me just start off with acknowledgements, because sometimes we run over time and we forget to acknowledge the people who do a lot of the work in the lab. So a lot of this work has been conducted by my PhD student, Alana Goldenkopf, and I have some great people that also helped out, a great undergraduate student, Danielle Destiny, great RA at the time, Katie Michon, who's now a PhD student with Thad Polk, and a postdoctoral fellow who's been helping with some of the imaging from Dr. Taraz Lee's lab. And again, all these people here. And as Roger already mentioned, a lot of great mentorship from Dr. Robert Chen. So let me just start off with giving you a little bit of an introduction to what I'm interested in. So I'm interested in how the brain controls behavior. And many scientists may approach this very large question by starting with perception, and then asking, how does the brain build an internal representation of the world, and how it then uses this representation to guide action. In contrast, what I do is I study behavior by starting with a concrete task, such as a voluntary movement. And I'll talk about upper limb movements, in particular, object goal-directed tasks, and asking what parameters of the task that the brain must specify and control, and what information from the environment it may employ towards that specification. So selecting and then specifying a goal. And that goal here is an understanding of brain mechanisms for mediating interactions with the world, not necessarily of the mechanisms for representing the world. So if you look at a research program like mine, what it's based on is on an approach that begins with the question concerning motor control, and then gradually works its way towards the perceptual system or sensory motor integration, which guide that control. So you could say that I'm kind of going backwards through the brain. So again, my research focuses on the neural control of visually guided hand actions, and I use a framework of prehension. So I feel that, and a bunch of us in our field feel that prehension remains a very powerful yet simple system for understanding these neural underpinnings of goal-directed behavior. And so in my research, the brain is seen as just like this information processing system that transforms sensory information into perceptual representations, and then uses this knowledge to construct an internal representation of not only one's body, but the world. And then we make some decisions on how we're going to finally implement these decisions through action. And so a lot of my PhD work is really focused on the parietal cortex. And I know when I say the parietal cortex, it's a pretty big area of the brain or region of the brain. It's comprised about 25% of the cortex. But I really started to focus on specific areas that have to do with reaching. And I've also done some work with eye movements and such. I won't go into great detail with that, but the general idea here is that we're going to construct this internal representation for action, and that this area is really, really important in both the integration and that sensory motor transformation. And so some people like Richard Anderson at Caltech have used these types of areas, or these areas, to decode motor intentions and use them in neuroprosthetics. Again, I'm not going to go into great detail here, just to give you a little bit of a background, because some of you don't know what kind of work I've done in the past. But a lot of the work, what I've done is looked at transcranial magnetic stimulation as a perturbing device or tool in where I can like interfere with functions. And by doing so, what I did in the past is I've actually looked at different subregions within the parietal cortex. So for example, the angular gyrus or the medial intraparietal sulcus, or an area that Jody Cullum at Western University in London, Ontario is called SPOC, which is just close to where the parietal occipital sulcus is, and really started to ascribe different functions to specific things, such as proprioception of the hand input, the visual input of the hand, and maybe sensory goal input. And from there, during my PhD, we constructed this model. And the take-home message here is that we have these overlapping systems. So you have your action, cognition, and perceptual system. And the main thing that I want you to take from this slide is that goal-directed grasping activates a parietal frontal network. So it's really important to recognize that the characteristic properties of brain area are not necessarily intrinsic to that area, but arise from its specific interactions with other nodes of the network. And so again, recently, advances in neuroimaging, in particular resting state neuroimaging, or even task-based functional connectivity, and a technique that I'll get to shortly called dual site transcranial magnetic stimulation. And sometimes I'll use terms like multifocal stimulation techniques. What they've allowed us to do is reveal functional interactions between these different areas of the network in humans for a specific behavior. And so much of the work that I'll talk about has to do with upper limb grasping. And so what this allows us to do is probe cortical-cortical connectivity between different areas of the network, and then in turn test whether these interactions are specific, in my specific case, to an upcoming hand movement. And so the idea here is that by using connectivity to identify the network, we can potentially better localize and even further down the road, treat neurological symptoms. So what are some of the implications of this network perspective on brain stimulation in improving and optimizing behavior? Well, what we do know is that the effects of stimulation can propagate beyond the stimulation site to impact a distributed set of connected brain regions. So a brain network shown here on the right. Although there is going to be local effects, so if we're looking at the motor system, most stimulation has been applied over the primary motor cortex and just looking at local effects. But if we start to zoom out and take this network-wide effect, we can see how different stimulation can modulate downstream and network activity. And so these results can support a network perspective towards understanding and treating these neurological disorders. And what this allows us to do is highlight the therapeutic potential for targeted brain network modulation. So more broadly, it's conceivable that these brain networks, and I'll show you some examples of this shortly, can be used to understand why brain stimulation works, and maybe further optimize and improve therapy by identifying both the best places to stimulate the brain and how we might approach stimulation application. Now, I'm not going to go into great detail here, but given the increasing evidence that these network effects are relevant to therapeutic response, again, it's possible that this non-invasive stimulation, so I'll focus on transcranial magnetic stimulation, to these different regions within a given circuit could have the same therapeutic effect. A lot of these questions are still ongoing in our lab, and we haven't addressed them, but we have some ongoing studies that look at, for example, this motor control or this grasping prehension network, and looking at whether stimulating, say, the parietal cortex or a premotor region versus the cerebellum have similar effects if we're stimulating the same network. I'm not going to go in great detail here, but here's just the basic conceptual research framework we have. But the take-home message here is that we're trying to understand the neural basis of motor or motor cognitive disorders and develop some new treatment interventions by understanding the impact of stimulation on motor and cognitive functions, and then using brain circuits that subserve, for example, these actions to guide the stimulation itself. And by doing so, we hope to inform and come up or develop better translational applications of the stimulation. So I think Ben touched upon this last month, but for those of you that weren't there, what we use is it's called TMS for short. So transcranial magnetic stimulation. And what we do here is we use a rapidly changing magnetic field to induce currents and action potentials in underlying brain tissue. Can people see my cursor when I'm moving it? Is that right? Okay, thanks. And so what happens is that if we, for example, place it over the hand representation of the motor cortex, we can elicit muscle twitches in specific hand muscles. More importantly, when it comes to therapeutic modulation, what we know is that we can use different repetitive patterns, and I won't go through them in great detail, but the one that we'll talk about today mainly is ITBS. It's called intermittent theta burst stimulation. And what we can do there is induce a long-term potentiation-like effect. So again, this can be used to manipulate brain activity. And what's important to recognize here is that the changes outlast the stimulation period itself. I'll talk about this briefly. It's a technique that only maybe a handful of labs in the world use, including ours. It's called, sometimes referred to as cortical paired associative stimulation. And what we do is we place two coils on a head, and we can modulate or strengthen the connectivity between the two. So it's kind of following Hebbian-like plasticity mechanisms, and I'll get into that shortly. So again, if we want to study the effects of transcranial magnetic stimulation, we can do this in several ways. So the way my lab will look at this is we can apply stimulation and then interrogate before and after the modulation what happens to motor cortical excitability, or more accurately, it would be cortical spinal excitability. And the way we do that is we deliver TMS, we get this motorable potential, and we can measure that with EMG, electromyography. A technique that I'll describe shortly is using this multifocal TMS. In the literature, sometimes it's referred to as dual coil or dual pulse or paired pulse with two coils stimulation. And what this allows us to do is interrogate the functional connectivity both while a person's at rest or while a person's performing a given action. And I'll show you an example of how we do that. So you can think of it as almost like a form of effective functional connectivity or effective connectivity in a given brain area, in this case, attached to a pathway that outputs to the motor cortex. And finally, we can look at the impact of stimulation, of this repetitive stimulation, using neural imaging. And I think there's some nice work that Taraz has shown in the past at these meetings. And Taraz-Lee, and again, looking at how we can modulate brain connectivity. And so we can use fMRI, resting state, or task-based connectivity, or EEG, for example. And some of the other techniques we do is we also just look at what's the impact it has on behavior. Now, some of the challenges or needs, shortcomings, if you will, of TMS-based clinical applications is that there's this high inter and intra-subject variability. In particular, when you're looking at something just as simple as motor evoked potentials before and after a repetitive protocol. And nonetheless, in addition to that, we also see that some of these interventions will have varying effects on behavioral responses. And an outstanding question is also, what's the extent to which some of these induced plastic changes can reshape long-term motor function? So we address this in several ways. So, and I'll get to some of the more recent projects that we've collected. And I'll talk about some of the ongoing work that we have in the lab. But most neural rehab, in particular in the motor system, has focused on targeting only one brain area. And it's usually the output because it's easy to find. And in this case, the M1, for those of you who don't know what the acronym is, it's primary motor cortex. The second shortcoming is that brain stimulation is usually applied while a subject is at rest. So, we're probing networks that are both inhibitory and excitatory. And again, there's fluctuations in signals. So I'll talk about state-dependent stimulation. And again, I'll get in more detail shortly. And finally, going back one, for the brain stimulation that's usually applied at rest, we're looking at if we can change endogenous brain states. And then the other thing is that you'll see for most of the therapies, there's really not, the dose is very low. And I'll get to that, and I'll borrow some principles that have been used in depression lately, particularly a protocol called SAINT. So again, the question that we're concerned here with is, can we boost the efficacy of repetitive stimulation on both brain and behavior outcomes? And so the idea here is, let's look at using a network-based stimulation protocol in order to change these functional network dynamics. And then I'll show you some evidence that we can actually change the brain state or control for the brain state using a behavioral task, and see how that may reduce variability. And then I'll tell you about some ongoing work we have about increasing the dose of stimulation, so exogenous stimulation. So much of the work, like I've mentioned, that's been done in the motor system will focus on a single area. So here, where are we going to stimulate? We're going to stimulate M1. I'll talk about shortly how we can change connections or strengthen connections using this cortical paired associative stimulation technique. And we can also use this, as I've alluded to, this dual site stimulation or multifocal stimulation to interrogate the way different areas talk to each other, if you will. But what I'm going to focus on the first study here is really what happens if we stimulate an area like the cerebellum. Okay, how does it affect the parietal cortex and M1? So obviously there's some direct connections to M1, but we can target specific areas of the cerebellum that don't have as many or very little connections to M1, but have more connections with the parietal cortex. And again, allowing us to see how we can get downstream effects. So that is going to lead me to the first study. But before I get to that, let me just explain to you how we can induce what we call motor associative plasticity using this cortical paired associative stimulation paradigm. So what we can do, I'm just showing you, because it's just easier to see, the coils over the left and right primary motor cortex. But what we would actually do is keep it on one side. So we'd have one coil over the motor cortex, and the second coil would be here at the back of the parietal cortex. And so we can deliver this at about a rate of 0.2 hertz. And we just do this for about 10 minutes or so. And what we find is that right after we finish that stimulation, we can increase the output at M1. And there's some other measurements that we do. And basically what this finding shows is that you can not only increase this corticospinal excitability, but strengthen some functional specific connectivity, in this case for a motor pathway. So we asked the question, or at least Lana asked the question, my PhD student who led this work, is can we alter distal motor associative plasticity in these parietal motor networks? And so the way we went about doing this is we got a functional scan, a resting state scan, and we identified the motor cortex, and then we found an area that in the parietal cortex that had the best connections, functional connectivity to that area. And then within that parietal region, what we did is we selected a region that was then connected to the cerebellum that had strong connections to the cerebellum, but very limited connections from the cerebellum to M1. So we really reduced that. We chose a target that had the least amount of connections to M1, but the greatest amount to the parietal cortex. And by doing so, what we wanted to do is look at what happens if we stimulate here. So we stimulate the cerebellum, which I'm showing you right over here. And then after stimulating the cerebellum, did this paired associative stimulation paradigm where we would strengthen the connection. So we would go back and we would see this increased activity or excitability. So, up until this point, most studies would actually just work over M1 and look at metaplastic effects, but not really assess how a more distal area can in this case, alter subsequent Hebbian-like plasticity mechanisms. So let me just show you what the data would look, or what the data looks like when we do that cortical paired associative stimulation. So we're looking at M1 output here after the fact. So after about 8 minutes of this cortical stimulation, so we're applying it on parietal to M1. And for those of you who are interested, I can give you more details later. We're using what's called an interstimulus interval of 5 seconds. So the stimulation subthreshold happens here at the parietal cortex, and then 5 milliseconds afterwards, we deliver a super threshold, so about a 1 millivolt stimulation that's elicited in the MEP. And what we see following post that cortical path intervention is that you get this increase in excitability, and it's quite consistent. We've repeated this in many studies now, where we get an effect that lasts 60 minutes or even longer. So then the question becomes, all right, now what happens if we stimulate beforehand using ITBS, so intermittent theta burst stimulation? Can we actually alter this subsequent Hebbian-like plasticity? So we apply it, and then right afterwards, 10 minutes afterwards, we start to interrogate and see what happens after that CPAS. And so here's what happens when we stimulate the cerebellum. So again, I want to remind you that we're applying stimulation for 3 minutes using this intermittent theta burst stimulation. Okay, and so the idea here is that it should cause a long-term potentiation. And so remember, every output coming out of the cerebellum is going to be inhibitory. So the idea here is that what we hypothesize is that we would probably cause an increase in that inhibition. And what you can see is immediately following that, so we apply the stimulation here, then for 8 minutes, we apply the CPAS stimulation in the parietal motor network, and then we do our measurements over here, which is reflected on the graph here, post CPAS. And what you can see that's quite striking is that we get a reduction in this parietal motor excitability that we saw before. I'm not going to have time to go through all of the control conditions, but we do various control conditions. So for example, we have an active control site over the visual cortex. And again, you do not, none of the control conditions will show that suppression, if you will, or reduction. So what this tells us is that that if we apply repetitive stimulation to the lateral cerebellum, it has distal modulatory effects on the parietal motor network. So this is the first study that we know of that's actually looking at causative data. So we're looking at, an objective measure here. Plasticity, we're defining it as changes in the motor cortex that are enduring. There's been previous studies that have looked at stimulation in the cerebellum and then using EEG or fMRI to quantify this. So they have some similar findings, but again, this is the first time it's been shown causally. And so this is just some work that's in prep. So I've just shown you how we can target distal areas to modulate activity. One of the other shortcomings or challenges has been that brain stimulation is usually applied while participants are at rest. And what do I mean by that? So what we find with a lot of repetitive stimulation, again, is that there's this increase in variability. And the idea here is that it's perhaps that the functional state alone, people are doing lots of things when you're stimulating them. And sometimes what that could lead to is ill-defined and spontaneous fluctuation that might render the TMS less efficient. So some of the things we need to consider, or many researchers in our field have considered, is looking at, for example, in training stimulation to local oscillatory activity. So again, they'll use EEG and MEG. Maybe using, and we've done some of this, getting some information from bowl signals, resting state, or task-based information either before or after stimulation. What I'll focus on here today is the experiment I'm going to show you now is actually constraining the brain state with a behavioral task. And in this case, the idea is that we can engage a subject in a task to prime the network of interest, and then make these neurons more susceptible to changes from stimulation. And what I kind of just showed you in the last one, we might be able to prime exogenously with stimulation. So, what I just showed you, where we stimulate the cerebellum and see how it may have some enduring effects to subsequent protocols. So how do we do this? What we do is we have a task that we've developed where we engage individuals in an object-directed grasping task. And let me show you how we do this. So let me go back to that multifocal TMS method. And in this case, rather than using it as a way of strengthening connectivity, I'm going to use it as a way to measure excitability or how these areas talk to each other. So it allows us to probe how these different brain regions, in this case, the parietal cortex and M1, interact with each other. And so what this allows us to do is provide direct and objective insight into cortical activity during behavior. And so here's how we do it. So we use what's called a test conditioning pulse paradigm. So it's a paired pulse paradigm, and we have the two coils. So let's just imagine that this coil here, the blue coil, is over the parietal cortex, and the black coil is over M1. And remember that if we apply a test stimulation over M1, what we can do is elicit this motor evoked potential, so this muscle twitch. And the way we do this is, in half the conditions, we deliver just a test pulse alone. And we set the output of the targeted hand muscle to be about 1 millivolt. In the other half of the conditions, what we'll do is we'll send a conditioning pulse. In this case, it's about 5 milliseconds, 5 to 8 milliseconds before we deliver the test pulse. And so what happens is the following. That MEP can stay the same, or that MEP could become bigger, which would indicate there's some type of facilitation, or that MEP could become smaller, which would indicate inhibition. So again, it's giving us this causal interaction between this two node circuit. And so what we've done, or what I've done actually in some work with Robert Chen in Toronto, is we've looked at, okay, let's make this state dependent. So I'm going to ask you to either grasp the top of an object, grasp the bottom of an object, or simply just touch it. And the idea here is that I can activate certain muscles that are involved in this and probe how the connectivity from at rest changes while planning. And we can get pretty good temporal resolution. So SOA here means just stimulus onset. So basically time after the go signal, and that's when the LED would extinguish. And we can then send these probes within these different time periods. And again, what it allows us to do, I'm just showing you some data here where I'm looking at FDI is just your index finger muscle here. And ADM, just think of it as your pinky. So when doing a grasp. The take-home message here is that this dotted line means there's no change at all. So it's interesting that at rest, this particular area that we were probing in the parietal cortex was inhibitory. But when planning an action, so again, this is before the movement takes place, you can see that there's an increase in the excitation. So there's a facilitation in these regions when planning either a reach or a reach to grasp movement. And if we were to go even more in depth, we can even look at when it's occurring. So it allows us to figure out where and when this happens based on state dependency. So again, this slide here is just summarizing that in that at rest, we have inhibition, and during an action or during the planning of that action or that action intent, we get a facilitation. So we can really, really start to quantify some of this state dependency that we have. So let's go back to that question. Can we manipulate the neural state of cortical grasping network during parietal TMS? And in particular, can it increase motor excitability and manual dexterity? So again, what we're going to do here is we're going to activate the cortical grasping network during parietal TMS while we're applying the theta burst stimulation, and in particular, the intermittent theta burst stimulation. And the idea or the prediction here is that if we have a more controlled state, or if we're activating, in this case, this blue region, this motor network, that those neurons will be more susceptible, and we would be targeting that network as opposed to just stimulating and just seeing what happens. And in a lot of cases, the stimulation may propagate to areas that are unwanted. So what I'm going to show you now is how we located where. Okay, so in the past experiment, I showed you how we can use functional neuroimaging or resting state neuroimaging to figure out where to stimulate. Here, we're going to use this multifocal TMS to guide where we're going to stimulate. And again, what we do here is we're using this paired pulse or this multifocal TMS while the individual is reaching the grasp. And as I showed you before, we're looking for the area here in the brain. So all we're doing here is we're placing a marker, which we're trying to do this without neuroimaging at this stage, because if we're going to put this into a clinical setting, we might not be able to always get images of individuals. And what we do is we use the 10-20 system, and this is going over P3. So it's an area over the angular gyrus. And then we build a grid with our neural navigation system that's about 1 centimeter apart. And then what I do is we move the black coil here to different areas while people are reaching, and we find the area that's personalized for that individual that gives us the most facilitation. And then what we'll do is we'll place the theta burst coil over that particular area. So again, what this allows us to do is to not just know where to go, but we can time it to when and how to do it, meaning we have them engaged in the task. And by doing so, we then apply the theta burst stimulation. So again, the idea here is now we've localized it. So we've targeted this cortical grasping network, and we're really interested in the parietal cortex because it's a higher order hub that's known to do the sensory motor integration and might be a more viable spot to target, in particular with stroke patients, because it's not, in most cases, is not damaged. And the idea here is that we can target the specific grasping network, change its functional network dynamics, and then in turn improve behavior. So just to go over this part again, when you're using theta burst stimulation, the idea is that we can increase excitability to strengthen these brain connections. And it has this potential for plasticity, and it's been shown time and time again that it may improve the induction of synaptic long-term potentiation and in turn improve behavior. So based on what we said there, the idea was, okay, if we engage the individual, we can target the specific grasp network. It's going to be a lot better than when they're just at rest. And what we did in this first experiment is we looked at what are the changes in motor cortical excitability. So we took those MEPs before and after each condition. And does it improve hand function? And so we used a gold standard clinical test where it's just a 9-hole pegboard test. And this is done in healthy individuals. A lot of this work was part of Danielle's undergraduate thesis with Alana, working together on it. And so again, we did our pre-assessments here. We also had a control task here. So just to make sure that this change, if there was any, was specific to the actual targeted cortical network, the prehension network, and not just some improvement in reaction time. So we had 3 conditions. We had the first condition, which was applying stimulation over the parietal cortex while they were engaged in a task. Just a note here, the stimulation takes about 3 minutes, so they're doing 3 minutes of reaching. But just before that, we actually have them do 6 minutes of reaching. So the idea here is that we were priming some of these neurons beforehand. We have an ITBS alone condition, so they're just watching the lights, and we're making sure they're paying attention. We'll ask them which lights turn on, but they're instructed to do nothing. And the third group is we go over an area of the parietal cortex that we've determined using that multifocal TMS technique to not be involved. So where we get no changes as a control site. So it's an active control site while they're grasping. And then we do these post-assessments that were similar to pre-assessments at up to 60 minutes afterwards. What I'm showing you here is the motor evoked potentials for each of the 3 conditions. So in blue, we have the state-dependent condition, ITBS plus grasp. In orange, we have the control condition where they're grasping, but it's over a different parietal region. And the other condition that was our main comparison is looking at stimulation alone. And what we do is we normalize MEPs to baseline, so everything is normalized to 1. And again, just to remind you, we set their initial stimulation at 1 millivolt, and this is 3 different groups. And what I'm showing you here now is that anything above this line, this dotted line here, would indicate facilitation. So you can clearly see here that only the ITBS group that was grasping while getting stimulation over the parietal cortex changed the motor output. So just a reminder here, what I'm showing you here is the motor evoked potentials over M1 following the parietal stimulation. So you get this increase in cortical excitability. All right, that's great, but what does that mean? Sorry, that's great. And for those of you in the neural modulation field, there's a lot of variability. What I'm showing you here, we had 8 subjects per group. This is at 30 minutes and 60 minutes. You can see it's very consistent across participants, whereas in the other 2 conditions, you don't see that. What happens though to manual dexterity? And this was actually really, really surprising to us because we'd expected that there would be a change, but this actually translated, this is what I'm showing you is normalized data, but this translated to about a 1 to 1.5 to 2 second improvement, which is just drastic. And again, these are healthy young individuals. And so again, only in the group where there was state dependency, we saw this increase. The other 2 groups did not show that. And again, if we look at the individual data, you can appreciate that there's very little, if no, variability. I think there's just 1 person here, and there was a slight improvement here, but at 60 minutes, it was consistent across all participants, which is again quite striking. And when we look at our control tasks, so it's not just a simple like they just got quicker or reaction time or movement time. It was very specific. We saw no changes in our control task. So does controlling behavior state during network target TMS actually modulate brain activity? The answer is yes. It seems to increase corticospinal excitability. It also improves dexterous hand function. So again, this is just summarizing what I just showed you. And so the take-home message here is that controlling the brain state by having someone engage in a task seems to increase cortical excitability and manual dexterity. And it seems to be very functionally specific when we're looking at these both behavioral and neurophysiological changes. So again, if we have to answer the question, can we boost the efficacy of RTMS on brain and motor outcomes in the motor system? Right now, I just want to say it's a preliminary yes. We have to get a bigger sample, and I'll show you some of the work that we're continuing to do. But it seems like this network targeted approach, where we're stimulating the cortical grasping network and then modulating or manipulating the state dependency that is engaging a subject in a behavioral task, seems to improve the after effects of stimulation. Okay. The other problem that a lot of studies have has to do with, and this is going to be really important when we start talking about some of the clinical applications, is that most studies will only have, as I showed you today, with healthy individuals, 1 session or 1 daily session if it's in a patient population. So if we look at some of the work that comes from depression, which I'll talk about shortly, this could be a big factor in why we see such variability, at least at the clinical level. But before I do that, this is with Steve Taylor, Thad Polk, and Taraz Lee. We're currently running an R21 where we're doing that brain state dependent protocol in 3 different groups and a lot more people. But we're also looking using a multimodal approach where we're assessing functional connectivity with fMRI, both a task-based activity. We're looking at the neurophysiology, as I showed you, with TMS and some more behavioral tests, not just the 9-hole pegboard test. And importantly, what we're looking at is what happens if we have 5 subsequent days of stimulation. So this could translate more to a therapeutic application. And so hopefully we can get a better understanding of the mechanisms, and this will allow us to develop some individualized neuromodulatory therapies with patients with neuro impairments. All right, so I'm just going to quickly, that's just some of the data I showed you. Let me see how we're on time. Okay, I'm just going to quickly talk about some work or some ideas, if you will, that Taraz Lee and I have been talking about, and I've mentioned some of this to Dr. Album. and how we might be able to use this type of therapy for persons with PD, in particular with persons with freezing of gait or some movement related issues that might be due to that sensory motor transformation, online control, or as Taraz will look at it from an attention motor or cognitive perspective. So we know that, as I've alluded to before, that repeated pulses or this repetitive stimulation can be delivered daily for multiple weeks, and that it can cause lasting changes in excitability. TMS has been FDA approved for depression, but not for PD. And so there's some large randomized control trials that show the benefit of motor symptoms and depression in persons with PD. And I can talk about this offline with people if they like. But unfortunately, there's been small trials that suggest that there may be a possible benefit for dyskinesias and freezing of gait. And this is important because we know a lot of the medications that PD patients take can result in those deficits or impairments. And so again, can we use this network-based approach, looking at where should we stimulate, and can we look at how maybe brain state dependent stimulation or dose, so how we apply it? Can this be, can these methods be utilized to possibly improve or optimize therapy? So I had mentioned before that there's some really, like the majority of the clinical work that's been done with TMS, because it's FDA approved, has been happening in the depression world. And recently there's been some nice work that's coming out of Stanford that uses this protocol. I think they call it SANT now, but originally they called it the Accelerated Saint Protocol, and then they came back with some rabbi protocol. But anyhow, and in this protocol, what they do is they use this intermittent theta burst stimulation. They increase the number of pulses. So the typical pulses are about 60 minutes. 600. But they do threefold. They actually do 1,800 pulses. And what they do is they use some novel treatment administration. So they use a 50-minute interval between the subsequent stimulations, and it's based on space motor learning. And they also target, use novel targeting in that they're more patient specific and looking at functional connectivity rather than just going to the frontal cortex, which has been traditionally what happens in clinics. And so the idea here is that we could possibly use a similar technique in Parkinson's patients. And what this allows us to do is apply, about 9 or 10 weeks of stimulation over 5 days. So some of the problems with previous studies that not only are they underdosed, but it's really hard for patients to be coming in for 3, 4 weeks, for 5 weeks at a time. So this may help. And as according to at least the depression literature, you'll get similar, if not better effects using this accelerated paradigm. I think most of us here, and again, we can have this discussion, will agree that, or at least the way I look at it, that Parkinson's disease as a system level disorder. So in the past, people were just focusing on the basal ganglia, but maybe we might want to take this new approach where we can look at how the cerebellum and different cortical areas interact, or maybe how they change or are altered, and how can we possibly intervene here at the cerebral cortex level or at the cerebellar level to offset some of the impairments that occur because of what's happening in the basal ganglia. So again, where do we stimulate? And so we can look at different patient characteristics to see if we can change these network dynamics and see how it can change interactions between these brain regions. So here's some work that I did with Robert. We didn't end up finishing it because Parkinson's Canada wouldn't allow me to bring the money over, unfortunately, and there was no one in the lab who could finish it when I came here to Michigan. But what I'm just showing you here is this idea that this is just off and on medication, how you can see how connections can change, or functional connectivity can change. So it can get increased or facilitation, off meds or on meds compared to off meds, and how that you get the inverse happening when you're looking at cerebellar and the basal ganglia. If we look at some nice work that was done by Giacomo Coccia's group in Italy, and we use this parietal to M1 connectivity, again, we see that there's an inhibition where we should be seeing facilitation. So again, this might allow us to select some treatment based on individual pathophysiological mechanisms. I didn't really have much time, or won't have much time, to go into these articles, but there's a lot of work, particularly coming out of Michael D. Fox, or not Michael Fox, his lab. And basically what they're showing is that, you can map some symptoms to brain networks, and they're using human connectome. You can look at, for example, the links between invasive and non-invasive brain stimulation. What they found, which is quite interesting, is that if there's an excitatory connection between the two, if the correlation between DBS is negative, you need excitation over the cortical areas, and that show a clinical effect, whereas the inverse happens if you have a positive correlation. And again, this is, so Steve Taylor was part of this, one of our collaborators. They've been looking at this distinct symptom-specific treatment for targets using the circuit-based neuromodulation and depression, and finding that's quite consistent time and time again is that regardless of where the lesions are or where the initial impairment happens, it's causing a widespread effect. And so Alfonso Fasano, when I was at Western, Toronto Western, showed that if you were to look at lesion sites, what they found time and time again is that there's an area in the cerebellum that was involved in walking that was disrupted, or the connectivity to it that was disrupted, regardless of where the lesion was in the individual. So that kind of just leads me to the project that Taraz, Lee, and I have been talking about, in that if we can just look at it from there being a sensory and visual processing overload. So again, looking at this new approach of looking at the basal ganglia, thalamus, cerebellum, cortex connections. I think that, a possible intervention based on some of the work that I've shown you and some of the ongoing work that we have is that we can possibly stimulate over the parietal region to target areas that are connected in the cerebellum and the basal ganglia. And there's some nice animal work that shows these connections done by Peter Strick. Another model could be looking, and this is an ongoing Parkinson's grant that Taraz Lee got an early career award that he's doing with Dr. Album and myself, where he's looking at the dorsolateral prefrontal cortex with this idea of this attention motor interface overload, and maybe target that area. And again, using functional connectivity to guide the stimulation. So he's now looking at, should we be using inhibitory or excitatory stimulation for that given area? I'm just trying to be cognizant of time here to leave some time for questions, and I'm happy to talk offline at any point in time. You can just send me an email and we can set up a time to talk. So that just concludes my talk here. Thank you.
Okay, thanks very much, Mike. Any questions? Let me ask you just a couple. First of all, I'll mention, actually, you may not know this, but Mike Fox is on our external advisory board for the Udall Center, actually. So
yeah, he was at Toronto Western when I was there, and I bumped into him once at the hallway, and that was my limited interaction with him.
Yeah. Well, he's been very productive. He's actually burning to come to Ann Arbor so he can go to an Ohio State game, since he's from Columbus, actually.
Oh, okay.
In any case, so one question though, so one obvious sort of state-related thing is obviously people who are on and off their DBS stimulation. I know Robert has done some work in this area, but what about using these methods to try and interrogate possible DBS mechanisms in terms of circuit alterations?
Yeah, so Robert's done a bunch of work where he looks at M1 connectivity, and you can do other different types of measurements using TMS to look at the neurophysiology. Some of the work that we were talking about doing at the time, and and at some point, I'll probably have to talk to a few people that are doing DBS here, is we can interrogate, for example, these parietal motor networks off and on stimulation. Interestingly, there was a study that Robert had just completed when I was just leaving his lab, where they were actually using, in a sense, like this multifocal stimulation, but he was pairing the stimulation with DBS as best he could, because obviously the stimulation goes on for a long time. And he was showing that he can get a facilitation over M1. So think of, like, instead of using 2 coils, the conditioning coil, if you will, would have been the DBS electrode. This gets a little messy because of the timing and such, but he had someone that was working on it, but he ended up going to NIH, so I don't think they followed through on it, because he's been working a lot now with TUS ultrasound, focused ultrasound. But yeah, that is a possibility, that we can interrogate how DBS can modulate these networks.
Okay, all right, excellent, thank you. Any other questions for Mike? I don't see any in the chat or anything.
Hey, Mike, I was going to ask
Cindy.
one real quick. Yeah, sorry, I've got a weird visual here.
Hi, Cindy.
So really interesting work. So one thing, I'm not actually that familiar with the functional connectivity and the stimulating aspect, and I was just curious as to whether the direction of connectivity, do we know anything about whether that matters and whether that might affect your design?
Yeah, when you're saying the connection, are we talking like resting state or task based, like in the scanner, or the other type?
So I mean, I guess you could look at either one, right? So like, I mean, if you take a look at some of Calhoun stuff, like on the dynamic connectivity, like you can start to make guesses even on resting state, but I guess the task would be more interesting.
Yeah, that's what we've been working on now. So that long-term study, we have a task that Taraz developed using a force pinch. Yeah, so it's interesting. So if you look at some of Michael Fox's work, the negative correlation or the anti-correlation, the effects might be, I think there was a paper, I haven't read it yet, but I remember him mentioning something at a talk where it may inform if you should be doing inhibitory type stimulation versus excitatory type stimulation. And so the work that Taraz is doing right now is really looking at that, where he's doing inhibitory and excitatory stimulation to see if there's a difference in the networks based on it. So it's under investigated, I'd say, at the moment.
Okay, thanks.
Yeah, but it is an interesting question, because I think that will matter, right? Like, because we know, for example, if I hold a contraction and I stimulate, I can get the opposite effect happening.
Right. Yeah. I mean, that's what I think. Like when we take a look at the fMRI, connectivity, it's just sort of like we just see blah everything. Although you can, as I said, you can try to tease apart some of the dynamics.
Yeah, and so that's what's nice about the dual site. The only problem is it works well with the motor system, because I need a motor output for that measure. Some people now are using TEPs, like TMS evoked potentials. So doing it with EEG, and I think some people are doing FNIRS, but I don't know enough about FNIRS to comment on it. So there's different methods. So what's nice about this dual site, though, is that we could, we can get like a causal, like there's definitely inhibition or facilitation, where sometimes with those correlations, it could be inhibitory and it's just positive, or inhibitory or anti-correlated, and that's positive too. We don't know.
Okay, thanks a lot.
Thank you.
Okay, excellent. Any other questions for Mike, or? Okay, well, I guess not then. Well, Mike, thanks very much for a very nice talk.
Yeah, thank you for having me.
Much appreciated. So I think